promoter repressor maintenance, that is PRM, and inactivates it. So this will stop the synthesis of C1 repressor. So once the C1 repressor is stopped, the transcription begins. Now the CRO protein also has a positive effect on the operator promoter left and operator promoter right. So the structural genes associated with these promoters, they start getting transcribed, and this will lead to the lytic development. So this covers the point of lysogeny and then the lysis, and we can see here how C1 repressor, when it is absent, there is no negative effect on the operator promoter left and right, and when CRO is present, there is a positive effect on the operators, and thus the lytic development begins. Now, some questions arise during this discussion that because the lambda has the option of both lysogeny and lysis, how does it decide, right? So there are some questions that need to be answered. The first question is that how does the infected cell, the infected bacterial cell, decide to become lysogenic rather than to enter the productive cycle, or that is the lytic cycle? The second question is how does the lysogenic condition get maintained into the infected cell? Now the answers to these questions depend on two things. The first is that is before we begin the discussion of the answers, we must understand that the infected cell undergoes a decision between the two alternative pathways, that is the lytic pathway or the lysogenic pathway. Now in those cells which are rich, that is sufficiently nutrients are present, then in such cells there is a high protease activity. This high protease activity leads to the breakdown of the C2. So this will lead to the lytic life cycle. Now in those cells with limited nutrients, the protease activity is low, making the C2 stable. So this will lead to the lysogenic lifestyle. So it is the condition of the nutrients that affects the lysogeny or the lytic. If there is high nutrients, then lytic. If there are low nutrients, then lysogeny. So if the lysogeny is followed, if the latter, that is lysogeny, is followed, the state becomes stable, it becomes established, it becomes maintained. While this state is highly stable, it can switch from the lytic path to the lytic pathway in the process of prophage induction that we have already seen earlier. Now this induction, as we know, is due to some UV exposure which occurs when due to the host SOS response, which is triggered by the DNA damage due to the UV exposure. So it is the UV exposure that converts the lysogenic state into the lytic state. Now this whole story is again put forward in a single flow diagram. How there is a bistable switch. This bistable switch consists of, as I mentioned earlier, the CRO and the C1. As you can see in the first line, that is, if the CRO is off and if the C1 is on, this will lead to the lysogeny. Or if the CRO is on and this C1 is off, this will lead to the lysis. So this covers the whole story of the lysogeny and the lysis. And with this we end the discussion and the references used during this discussion, that is a very good book from A Genetic Switch, Gene Control and Phage Lambda from Tashne, The General Virology by Luria, Campbell and Bartimol Darnell, and the last, that is Brock Biology of Microorganisms by Medigan, MT, Martinko JM and Parker J. So this completes our discussion of regulation of the lysogeny. And thus, I hope the viewers are now clear with the regulation of lysogeny. If there are any questions from the viewers, we can all discuss that and we can continue our discussion for a few minutes.